Welcome to Mass Maritime Academy, and in particular to our research labs and research vessels here on the edge of the canal. My name is Fran Veale, and I'm the chairman of the Marine Science, Safety, and Environmental Protection Department. Today, we're going to show you a video that will be showing you some of the equipment and technologies that MSSEP students use in their research. Students at MMA participate in a wide variety of research programs, in particular water quality, bottom sampling, nutrient analysis, as well as hydroponic studies. All these areas are critical to understanding and protecting our environment. Today, the ship is heading from MMA to monitoring station number 9. There are 2 ships that we're using, both of our research vessels. The first is the research vessel, the MMA Zostra, which is a 22-foot center console, and it's called the Zostra. And then we have Camryn, which is a 42-foot twin diesel. On board the Zostra is Dr. Kevin Hefferan and juniors Camryn, Roberts, and Tegan Murray. And the vessel is being piloted by Bill Klimm, director of the Marine Department. On board the Camryn is Professor Bill Hubbard and seniors Jack Gerrier and Courtney Toomey. The Camryn is piloted by John Edwards. And a very special thanks to the Marine Department, who has put significant effort into developing this research program. Not only have they been instrumental in finding us vessels, but in out-equipping them and ensuring that that when we do go offshore or go out into the bay, that they will captain the boat and make sure that the best of seamanship is being used. So we really want to thank them for their efforts and so forth. So at MMA station number 9, we'll all review the technologies that are generally used by the students. Now, I realize that it's probably a lot better with hands-on and being able to get out there, but due to the circumstances we're presently encountering, namely the pandemic, then I think we're going to have to wait a little. We are still taking students out, but we're doing it more on a limited basis. So, in any event, I think you'll be hearing from Professors Hubbard and Hefferan on different techniques that are being used. And hopefully, in the very near future, you'll be able to participate and be using these techniques. Some researchers will decide to go that way.
Yeah, we should have told you that. Yeah, it wasn't meters, sorry.
They told me that afterwards. I'm like, what? I'm going to go back to this number.
Okay, please nothing because you guys never have. Is that tight or something?
What the eye?
Got it. All right, so we're going to start with Jack setting up his small trap here. We're going to go down and see if we can get some juvenile black sea bass. The black sea bass study going on at Mass Maritime needs some juveniles in the 4 to 5 inch range. But even if we get younger the year, let's say 2 or 3 inches, and we're using the scientifically proven Doritos bait, and honestly that is the best bait for...
Do we have the line? For the
line coming up.
How about we put some bait in the water?
Yeah, bait. Got donuts. He's using Doritos. This is the best bait for juvenile fish, to say the truth.
Not only a bait for little fish.
So we're going to start with the water quality profile. This is your YSI Pro DSS. You can see that screen there, and I'll go through it a little. But we're able to do a profile, and it gives you temperature, salinity, dissolved oxygen, depth. This one even does pH and total suspended solids. Today we're just occupying Station 7 here off of Mass Maritime Academy, right outside of Onset. And we're going to just do a profile at every meter from top to bottom. Courtney is going to record, and what will wind up is a nice three-dimensional picture of the water quality. The best thing to do, the best thing to do before you go out to sea, is have a data sheet. Even if you don't fill in everything on the data sheet, at least you remember what we're out here to get. And it does remind me that we have to get pH and we have to get a SETG reading. So let's start with the profile. To do that, I'll put this through. This is the probe. Always keep the probe in a little moist water. It's about 100-foot capacity here. So just tightening. This is a probe guard, and as you can see, there's 4 or 5 different individual probes that are replaceable. So this is the marine complex of them. Temperature, salinity, dissolved oxygen is what we're most concerned with. So the first thing to do is let's start at 1 meter. And I'll read to you, and I think what we'll do is just, while we're going on to something else, Courtney and I will sit here and get the data throughout the water column. And it takes a minute. Depth always takes a minute, but let me read through what we've got for conditions. Depth is still jumping around. See, the depth says 0.2 of a meter, and I know we're down a lot farther, so there we go. Depth meter woke up. So at 1 meter, we have 16.7 degrees centigrade. Easy way to go centigrade to Fahrenheit, everyone should know this. 16.7, let's call it 17. Double it, that's 34. At 30, the water temperature is around 64 degrees Fahrenheit. So for October, that even means the lobsters haven't come in yet. Lobsters don't really come in until about 65 or below. So we're at 64, and so it's going to take a little more. The dissolved oxygen is at 7.73 parts per million. So parts per million, or milligrams per liter. So it's dissolved oxygen. There's also a reading on here of percentage. And the percentage of oxygen, the computer automatically calculates whether or not we have the most oxygen you can or more dissolved in the water. That seems odd, or more. If there's a lot of photosynthesis, as in late afternoon, you'll get a supersaturation of the oxygen. But it's all dependent on temperature. If there's no addition of oxygen and the temperature's constant, you can get 100% saturation at different temperatures. So if the water temperature is warmer, it'll have a lower amount of oxygen. If it's colder, it'll have a higher amount of oxygen. So calculating the temperature is good, because then you know percentage of dissolved oxygen. Is the water healthy? And that's what we're looking at there. Oh, more data? Yeah, Courtney's saying, more data, please. And dissolved oxygen, and most of us know anything over 5 parts per million is okay. Dissolved oxygen, 3 to 5, less than 3 parts per million of dissolved oxygen, EPA characterizes that as impaired waters. So we're good, healthy water. Salinity, 32.08 parts per thousand. So we had parts per million in dissolved oxygen, parts per thousand. You know, don't forget, let's sit down with a pencil and make decibels one of these days. 0 point, tens, hundreds, thousands, 10,000, 100,000. So understand what parts per thousand is. You know what parts per hundred is. That's percent. That's money. But the salinity at 32. It's pretty high salinity, to tell you the truth. We haven't had a lot of rain. We're going to have some rain in the next few days, it looks like. The 28 to 32 is estuarine salinity. You'll probably see at a coral reef something up around 34. And I don't know, there are a couple of the Red Seas, and I'm looking at Professor Hefferan. Aren't they a little higher, the Red Seas? Yeah, some of the more tropical. Yeah, but we're 28 to 32 is just normal. We like normal, by the way. And we're gonna just stop here with that. There is pH, there's total suspended solids, but what we want to do is get a depth profile. By the way, this gives you latitude and longitude. It is GPS computer. Also, besides, we like to have a hard copy, but also if I push enter, what'll happen is it'll record all of this data, and this data will be recorded in an Excel format. So this is really the technology that we're getting at now, that everything is just quickly transmittable. And there's a download cable, the port's there. Of course, we lost the cover to the port. But the next generation is instantly downloadable to your cell phone. So that's the technology. Yellow Springs instrument, YSI technology. There's different sizes of these. There's ones that just do salinity and temperature, but this is the industry standard. Okay, good.
Camryn has a video camera that she's lowering into the water shortly using the davit. And she's using a rope, and there's also a gray wire attached, and the rope allows her to go down to 4.8 meters depth. And then we have a screen that she's going to tell us what she sees on the bottom. So the screen is going to demonstrate, may not be very visible here. To demonstrate the substrate and organisms that are both on the seafloor as well as critters floating by. So Camryn, what do you see?
So right now we see a sand substrate and a few different fish and some crabs. Other than that, that's pretty much all we're seeing right now. We have some dead seaweed and some shells as well.
And this summer, Camryn was involved in a group that was monitoring eelgrass throughout Buzzards Bay. So we used this to determine the percent or presence of eelgrass, percent density, percent epiphyte that was growing on the eelgrass, as well as substrate, depth, and the presence of algae. So eelgrass is an indicator of good water quality, and the Department of Environmental Protection of Massachusetts is working with us to monitor the entire shoreline of Massachusetts in order to gauge water quality over this 4-year period. So we mapped the south side of Cape Cod, this year Buzzards Bay, next year the north side of Cape Cod, thereafter up to New Hampshire. And it'll be a continuing 4-year cycle. And Mass Maritime students such as Camryn and Grace Jensen have been participating in this in the summertime. And we will continue to do that through October when the eelgrass dies off. Anything else you see in there, Camryn?
Right now it looks like we have some eelgrass blades. If you look closely.
And it may not show up well on this monitor, but
Yeah.
Camryn has a better view. And eelgrass is a good sign again of fine water quality. Thank you. The camera now is demonstrating the GTEC.
So this is our GTEC computer, and we are on Arcgis Collector. And all these green points are points that we've already completed. And so this is our location right here. to show how we complete a point, you select the point type, and the first thing you want to do is go to the camera and you want to snap a photo. Usually we're looking for eelgrass. There's nothing in this photo, but we're just going to take it anyway. And then we list if eelgrass is present, so Zostra. So we don't have any eelgrass, so we're going to say false. So the density would also be none, since there isn't any. There's not going to be a location, because again, we don't have any eelgrass. The epiphytes are what's on the eelgrass, so we would just list none. And then algae present, it looks like there was some in the photo, so we would say true. And then algae type, it's going to be mixed. And then we go for the density of the algae, so I would say less than 5%. And then we had that sand substrate again, so we'd select sand, and then go for the depth, and we read that off the SIMRAD, so 4.8.
Let's drop a point here actually.
Yep. And then sometimes we add observations, such as if the eelgrass bed is patchy, we'd list that, or if we couldn't directly reach the location because it was too shallow. But after we were done with that, we just tap on our location, and then we just submit it. So when we submit that, that goes to Mass DEP. And again, these are all the points that we've completed this summer. So we mostly did around Woods Hole in Martha's Vineyard, and even around Buzzards Bay.
And DEP has aerial imagery flown. We hoped in May and June, but sometimes in July. And based upon that imagery in the field, we test where they believe eelgrass is. In their offices, they are drawing polygons, so the green lines are polygons that represent areas they believe eelgrass exists. And we go out in the field then, and we field check and verify the existence or non-existence of eelgrass. A lot of times algae such as codium will give the same spectral signal as eelgrass. So our job is to field test what is observed in imagery, which is flown in the summertime. And we continue working on this through October, and again, a 4-year project.
Alright, so just some equipment on the boat. Here we have the SIMRAD, and on here we can track our depth, the substrate, and we'd also type in our location, and we could follow the path to where we are going. And we also have a jack plate that we can move up and down, so we can use this switch here. So you can move the whole motor up or down, and this is very helpful because most of the eelgrass locations are in very shallow areas. And then, so our motor is 150 horsepower, and these are just some of the switches that we give electricity to. And up here, this is where we have the inverter, and we have where we record all of our footage. We have a USB in there for our footage of the underwater camera. And this here, when it's red, that means we are recording, so we are recording right now. This is one of our monitor screens. Usually the person operating the camera will be looking at this screen, so they can help the person on the GTAC. And then the person on the GTAC is usually going to be up at that front seat, and there's another little screen that they can look at. So again, you just sit right here, and you can just, so you can fill in the data.
So a very successful summer. This is our first year using a 22-foot boat, and we anticipate doing so in subsequent years. And each year we're bringing 2 students who serve as co-ops, 240-hour co-ops, and they are doing summer work. It's paid, and it's a great experience to get out in the field, to explore the waters off Cape Cod in Massachusetts, and conduct important science that monitors water quality. And here's Tegan and Camryn, and they're both skilled in working with boats and scientific equipment, and a great asset to our program. Thank you.
So Jack went overboard and picked up a couple local species. These are Labinia emarginata, and you would count the spikes along the back to see if it's Labinia dubia or Labinia emarginata. So if you took a marine invertebrate zoology, that would be the first thing you have to learn. but it's a spider crab. It is the relative or to the same family of the king crabs and the Veale crabs that you see in Alaska, but the meat content here isn't all that good. They do pinch. They're annoying little things that will pinch. And they do hang on. He's just hanging on like, why are you doing this to me? Now I'm covered with crab mud. So also we got a few other local animals came up.
I'm not really sure what the name is.
So there's a nice
Shrimp
little
species.
grass shrimp in here. Praying down. There's 2 species of local shrimp, and they really probably one of the most important organisms here because they take, yeah, it looks like a grass shrimp actually, Paleomenides. So they take primary productivity. They take the broken down plant material and they'll change it into animal material, basically shrimp. We also have a couple of our local favorite fish. You see a 2 little crabs there. That actually looks like the Asian shore crab. And
Oh no.
no. Okay, is it a good baby green crab, or it could be a? I don't have a microscope out here, so it looks like it's a black clawed mud crab. Yes, it is. Yeah. Those nice little black claws there, so that's a black clawed mud crab. Let's jump in the water and see if we can get something that shows us what this fish is. It looks like a local fish. So Courtney set up a hydrophone, and you could hear without any problems. You could certainly hear the tidal current going by the boat. So that's what we think you're looking at right now.
So we're doing underwater acoustics, underwater noise pollution studies in the Cape Cod Canal. It's heavily transited by recreational boats, so many different types of shipping vessels and tugboats, and this creates a noisy environment for marine animals that live under there, and it really creates confusion with them. So underwater noise pollution really interferes with all of their daily activities like feeding, foraging, navigation, finding a mate, and all these other different types of things. So underwater noise pollution is a huge issue that the scientific community is just starting to really get into and really understand. Here at Maritime we have some really awesome state-of-the-art technology that lets us listen under the water and lets us hear what kind of is going on beneath the surface of the waves. Today is a quiet day, so we don't get too much interference with the wave water ocean air interface, so it's pretty quiet today. You can hear a lot of crackling and stuff. That's the crackling shrimp that we have at the bottom. This right here is our recording device. We can plug this right into our audio converter that brings it in from our hydrophone. Do you want me to pull up the hydrophone so they can see it? So here is our hydrophone. It has a weight on it, so just so that it'll sink all the way down to the bottom, because it is just a cord. We have it secured to this line here, and then drop it overboard. and secure the line to the boat. We use this data on our computers and we bring it in using software called Raven Light. It's from Cornell University. It's actually really a cool program. It allows us to make spectrographs of all of our sound data, and we can actually visualize what the sound looks like underneath the water. And through this we can use different programs to analyze the frequencies and the amplitude of the sound and compare that to thresholds of anthropogenic noise pollution and damage to marine mammal hearing, or interference with their navigation and communication. And see what's happening in the canal. So harmful vessel noise in the Cape Cod Canal is part of Massachusetts Maritime Academy's environmental monitoring study for senior cadets. I've been doing this study for a year and a half now. This is my third semester working on this. Now it's my independent study. One day I hope to get my master's or PhD in underwater acoustics. I'm really interested in whale ears and noise, so this is a great project to learn the equipment and the technology.
Hey, we're going to trip this right now. I'll drop it here.
Yeah.
So the door is closed and we can catch a water sample. I have the top attached, pull out the bottom. Now both doors are open, top and bottom. We can lower the water sample into the water. This is our messenger. This trips the trap, essentially catching the mouse and the bait in the trap. But here the mouse is water to get a sample. Okay? Put it on the davit. Lower it down. And now I'm going to drop the messenger and close the door. And we'll get a water sample. We'll pull it back up. And we have collected successfully a water sample.
All right.
We'll bring that over.
So this is a Van Doren. There's a whole mess of different types of these water bottles, but it is the standard for the industry. We're going to do some analysis on this. We're going to start first with pH.
Here we have a Secchi disc, and this is one of the tools that scientists use to determine water quality and visibility in the water column. And the way to use this device is to let it down until it reaches a depth where you can no longer see it, and then at that point you'll be able to determine the clarity of the water that you're sampling from or doing tests on. So, down over the side. Just about there. All right. Now I can no longer see the white of the disc, so I know that I'm right at the depth where visibility ends.
Oh, we got our water sample, and although we could be a little neater, there's a nice little valve and you could drain the water slowly out of the Van Doren. I have little patience, so what I usually do is get it on my feet. What we want to do, and everyone else is right, what we want to do is just rinse the containers we're going to use with some sample water. This is a 1 meter sample. We'll call this one trash right here. We need a little trash water. And we're going to do a couple things. First, we're going to start with pH. And although the YSI has a pH meter, it's sometimes more accurate to use this type of just handheld laboratory pH meter that comes out in the field until it rusts. And we should be all set. I'm going to rinse the probe in some sample water, and then we're going to take a pH, and on the pH meter, it's an interesting thing for the pH meter that it'll adjust until you see a little smiley face. Do you see a smiley face yet, Tegan?
Yes, I do.
All right, so we have a smiley face. That means we have a stable reading. So we have a stable pH reading. We're going to enter it onto our data sheet. What do you have for a pH?
8.08.
So less than 8.1. So 8.08. And the typical estuarine pH is 7.9 to 8.1. If you're at a oceanic site, you'll probably see a little higher pH. I heard coral reefs are 8.4. I don't know, maybe some of the other oceans that have a little more salinity would carry a little more pH also. So we have a good accurate pH reading, and I'm just going to put the probe back in here, and maybe I could show you what the screen looks like for pH reading. Now that we've got our water samples on deck, we're going to use hot colorimeters, field spectrophotometers. They're one step up from a colorimeter. And what this item does is it puts a series of light wavelengths through your sample, and it records what that wavelength is and converts it to the amount of phosphates or nitrates after a chemical reaction in the sample vial. So we're going to do a chemical reaction, and we're going to record the intensity of the reaction, essentially how dark the liquid gets based on that reaction, and that'll be our nitrate or phosphate determination. So first what we want to do is get blank, because if this water is turbid, and it looks beautiful today, but there is some turbidity in it, we want to adjust the spectrophotometer to the water turbidity. So why don't you folks get the 10 millimeters to the meniscus. Go ahead. Then we need to just do a quick sample wipe. The soft cloth, honestly I usually use my sleeve, but we will use a soft cloth. And without touching the glass, put it into there with the diamond facing the diamond. See that diamond in front? Push it down. So once you clean it, don't touch the glass, and then cover it. Only gets one way. We want to go like this. We want to put it diamond facing diamond. And hold the cover kind of tight because you don't want any excess light. So then you want to zero it out, and that's the blue button. Just hit the blue button, and it basically goes dash, 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 and then it turns to 0. So what we have is compensation for whatever interference might be in the water. Now it's time to make a chemical reaction. So now that it's zeroed out, pull out those sample vials, and for phosphate, you want to tear this and put that one. It's a pre-packaged chemical reaction. Pre-packaged chemical reaction, and well, I'm glad you did that. I can never open those. The process says that she's going to shake it for 20 seconds, then wait exactly 2 minutes. And in 2 minutes, we can open that. We'll just time 2 minutes. Then you'll take your analysis. So go ahead and put it in. Shake it a little harder. You got it. Phosphates will go completely. And that's good. Okay, so let that sit. Put it right in here and close it. Just now we want to time that for 2 minutes. For nitrates is an extremely difficult thing to do, especially in the estuary. So the hot chemical company has put together a vacuum packed chemical reaction in a breakaway vial, and I'll do it just because I usually get cut, so there'll be a drop of blood. So basically you put it underwater, and the vacuum will take exactly 10 milliliters of water and put it in to the reaction, and then you're going to shake it for 1 minute and 20 seconds. So let me get to the reaction, and so I break it underwater, which is why we have that waste there, because then you're going, so now it fills exactly to 10 milliliters. Put that on it and shake vigorously, don't get cut, for 1 minute and 20 seconds. $9.50. And that's 2 minutes. So go ahead, and the green check button is your number. And what did you get for a phosphate number?
0.21.
0.2, so excellent. And let's see if we can show you what the screen looks like. Simple enough. There's our reaction, there's the number, and it gives you an on-the-spot reading of phosphates in milligrams per liter. So again, parts per million. You're good shaking. So now we're going to time the nitrates for 5 minutes. It's optically perfect glass, so that could go in. And in 5 minutes, 4 minutes and change, we will start our, we just hit the blue check button, and that's how long it takes. And we have nitrates in milligrams per liter at 0.8. Now, honestly, there's a conversion factor we have to do back on land with temperature, but it gives us this reading in the field, and it's easy enough in the spreadsheet to do the calculation. Relatively low nitrates and low phosphates. Veale benthic grab. It's going to sample at the sediment water interface. It's going to take a sample of what it lives on the bottom down about 5 to 8 centimeters. The Van Veale is 1/25th of a square meter, and so then if there were 4 clams in this sample, we could estimate there are 1/25th, 4 times 25, 100 clams per square meter. So you get a quantitative definition of what lives on the bottom. And it's not, we saw some crabs before, and we saw some shrimp, but it's not always that. A lot of times it's small worms, but it's all important food, especially right now this time of year for juvenile black sea bass and juvenile flounder. So it's nothing more than what looks like a big, you see this, a big dredge, or just a big bucket you see for a construction site. We can look at the top when it comes up. We can see what it looks like, and we usually stabilize the other one so that it doesn't open through the water column. So you only need 2 or 3 hands to set this, but it usually after a while you get the hang of it. So you have to hold it a little bit stiff and catch both of these pegs right there. Weighs about, I think this one weighs about 35 pounds. Which is why Jack's going to pull it up and I'm going to put it down. So I got it. So what you have to do is just feel for, and why don't you pull your trap out of the way? I think I'm just about to hit your trap. Just pull it to the right. So we have a trap down. We're just going to move that a little. It's clear water, so I can actually see about 10 feet down. I can see your trap. So we're going to go down to the bottom, slowly. Let go and then pull up. You don't want to go fast because if you go too fast, it'll actually wind up on its side. And we're going to go into that blue bucket that has a screen bucket with a 500 micron seed. All right, 500 microns. A micron is 1,000th of a milliliter. A thousandth of a millimeter. A meter, broken into 1,000 pieces, is a millimeter. So 1,000 times more than that. So that's microscopic. This is 500 microns, okay? 500 is a half of 1,000. So this is a half a millimeter screen in the bucket. What we're interested in is what lives in the substrate. And I'll bring in a close camera. And we can see there is a sand, it looks like a silty sand, it is. But you can see some worms living on top of the substrate. You can see my favorite animal, a crepidula. But now we have mud. So what we want to do is screen away the mud. And to do that, I'm just going to open the grab. And you can see, that's good. You can get it. Jack is well adept at mud. But you can see there's a white layer on top. So that is an oxic layer. It's called the RPD, redox potential discontinuity. I think you need to know that in this world. But it tells you that there are worms, and there's one right there, reworking the dark mud and putting oxygen into it. And that's what benthic organisms do. Does that look good? Would you mind doing the honors of doing a quick screen on that?
Sure.
So Jack's going to take and give you a little more water. You could do this overboard, but it would work. That annoyed the crabs, but that's okay. All right. So actually, we got a pretty productive bottom. I'm gonna clear that, and you can see. Why don't you look at the screen there for a minute? Again, 500 microns, a half a millimeter. One of my favorite animals, Crepidula fornicata. So this is a snail, and you've seen these snails. They look like slipper shells. And we've done some studies, Maritime published on Crepidula beds, and they'll stack up for the winter, and you can see a curl right there. And we have found 7 places offshore of here where the entire bottom is covered with these, and they're 100, 150 acres. Science pilot here, John Edwards, and I usually go out 2 or 3 times a year with students, and we've been monitoring them, and sometimes we get out there and the bed's wiped out. Other times they start looking like this, where they're just forming the bed. So this is a pretty productive bottom. You're also seeing worm tubes. And I don't know if you can see it, but there's a little worm sticking out of that tube. So if you're a flounder or a nice hungry black sea bass juvenile, this is a really good piece of food for you right here, sticking out of the water. Again, they take the detritus, the plant material, and they will change it into basically an animal material, and you're at the bottom, you're starting the bottom of the food chain. Just gonna kind of shake out. So now what we would typically do on something like this for long-term studies, we would take all these organisms that are on the screen, we would very carefully screen this and decant it, and we would put these organisms in formalin, and then we turn them into the lab, and you'd sit there in a microscope and you process, and we do this in marine invertebrate zoology, you process the sample and you count every organism that lives here. And I always say, if we count the organisms in your backyard and you have bunny rabbits and squirrels, and I have cockroaches and rats, then your backyard's a little more productive than mine. So basically, there's 4 or 5 other small worms here. Everything in here now to identify, we'd have to have under the microscope. But you can see, I think the point here is the bottom's not dead. We have quite a few organisms living down there. And if that's 1/25th of a square meter, and a quick count is we've got about 30 crepidula, you know, you have a very dense, set of organisms. Good eye, Jack. So that, this could be a snail, or is that a, no, that is a small hermit crab. So it looks like it's a Pagurus longicarpus. So that is a baby hermit crab. That's about as small as they get. So probably just hatched, and it used someone's snail shell. Again, these are kind of fascinating to look through in a microscope. That's that mud crab we caught before. I wouldn't be too surprised if we looked on this and found some other worms. And usually there's a different snail or a different organism, a gastropod or two. But we might just have an area that's worms.
And
So Jack's pulling the trap up. He just pulled up the trap. And again, it didn't have much. It was down for a little while. But we've gotten baby juvenile black sea bass. Well, I guess baby juvenile is redundant.
Yeah, that's a little tiny.
Yes, sea bass, yep. Well,
He's not doing so.
well, he's not doing so hot, so we'll start with him. So we're studying juvenile sea bass because prior to 1972, there were no juvenile sea bass north of Montauk. So Mass Maritime in particular can look at Buzzards Bay and say, well, the capitular beds are out there, shell beds have changed, and now we have this new species of sea bass in the last 20 years, and they're small. They're just this one is, they have a stripe on their side, and they'll keep that stripe until they're about 3 to 5 inches, and they're benthic. They live on the bottom and eat all those organisms we just pulled up in the screen. And then when they get a little bigger, they'll be able to school and move through and eat slightly larger organisms in the water column. Let's see if I can get this one without dropping him. But you can see it's a nice fish. See the black lateral line? So that tells you it's juvenile. That one is probably 3 weeks old. This one is probably 3 or 4 weeks old, sometime in September. And then by the time October ends, we're hoping to be big enough, and it'll be big enough to migrate and head south. Well, I say south, but just head to warmer waters offshore, since our waters will get down to pretty freezing. We have these in the lab at Mass Maritime, so we've been tagging and releasing. We're taking this back. We're trying to have about 30 of these, and we're doing growth experiments with high protein fish food. And NOAA has done one of those experiments, and it looks like this could be an organism for future aquaculture. So we'll keep him alive, of all things coming back.
hey, we just hauled the plankton net.
Got it.
And we unscrewed it, and we put this cap back on. And we have plankton stored here from trawling with this on net right offshore here in Onset.
So we take the sample, but if you look, you'll see thousands and thousands of organisms in there. Looks like we've got a good amount of copepods in there, juvenile species. We'd have to get under a microscope to give you a good look. You see, is that picking it up okay?
Lots of.
Yeah, give me a splash in here. Let me see if I can figure out what they are. And see if we can get the camera in there.
Lots of comb jellies.
You got ctenophores in there, so there's a couple comb jellies. Nemiopsis, a ctenophore, which is not a real jelly. It is a different jelly.
Okay.
I think. There we go. So you can see it's the clear jellies. You see ctenophore comb jellies, they're called. Right now we have the red purplish ones along shore, lion's manes. We also have sea nettle. They're a little fancier. They have thinner. And yeah, that's a good picture. Let's put it on the weight.
You have another tray?
Just go right there. Put it right on top of it. There you go. There's a good picture. And as the boat rocks, if you do this, you could get on the light, I should be able to cut some video and give you some of the smaller organisms that are there. And then you could see the ctenophore, the comb jelly.
Depth. And then we have a screen that she's going to tell us what she sees on the bottom. So the screen is going to demonstrate, may not be very visible here.
Let's take a few minutes here and just watch the underwater video. We've been running this while we were doing the other sampling. And notice you've got latitude and longitude on the left, and that changes within centimeters. It's the accurate system. And on the right, the date, the time is universal time code. It's GPS satellite time. So with daylight savings time, you'd subtract 4 hours. Then the bottom right number is the course over ground, so it's the compass heading of the vessel. and we're just at anchor and drifting around the same area to be able to do the topside work. You may have to turn your volume up, and if you turn your volume up for a few minutes, you'll hear the hydrophone recordings done at the same time as the underwater video. The juvenile black sea bass and basically any demersal fish see that one single laser pointer we have off the camera as a small crab or something darting on the bottom, and it's kind of entertaining. And we'll zoom in a minute, but it's kind of entertaining to watch them try to find that. It's like playing with your cat with a laser pointer on the floor.
So Maritime is very fortunate, particularly in the Marine Science Department, to have great Marine aid who gets us boats. So these individuals, John and Bill, allow us to do our field work. Without them, this would be impossible. So they keep our boats running. Even when mistakes happen in the field, they're able to fix us when knuckleheads get in the way sometimes. And they're always solving problems and helping us out. And we appreciate them greatly.
So, Captain and crew, say goodbye. This is brought to you by the Mass Maritime Production Company of Ocean's 11 here. All right, we're good for the day.
You can pick that line up. Go down underneath there.
Alrighty.
We'll go around the other side. The guy left.
We're going around.
What video? We'll go around. That?
Well, I hope you've enjoyed this video. Certainly, there will be opportunities for you after COVID to get out on the water. But in the meantime, it's a good idea to try to learn these technologies on land so they become a lot more simplistic when you get out to sea. And the one thing that I'd ask you to do is to think about the research areas that you want to work on over the next few years and ask faculty to help you, start a project. There are many faculty members here who are willing to work with you. And many times the research could even be your co-op. So I hope again that you've learned a little bit. In past years, the Marine Department and Bill, Kevin have taken the students out, and every single student had an opportunity to be out on the water. But unfortunately, that's not the case this year. But let's hope as soon as this pandemic breaks that you'll be able to get back out there. Well, thank you.